Well, biochemistry students, I now continue the concluding second half of the video presentation that we have on our Chapter 19 discussion on harvesting electrons from the citric acid cycle. We left off talking about step 4 of the citric acid cycle, in which alpha-ketoglutarate, shown here, which possesses 5 carbons in its chain, is converted to succinyl-CoA, which only has 4 carbons, through the action of the enzyme alpha-ketoglutarate dehydrogenase complex. After that step has occurred, we move on to step 5. Now we should notice that succinyl-CoA, shown here, is a thioester, which once again means that there is a sulfur-carbon single bond between the carbonyl carbon and this sulfur in coenzyme A. Now hydrolyzing thioesters is very exergonic, with a delta G value of negative 33.5 kilojoules per mole, which is comparable to hydrolyzing ATP. Thus, in step 5, succinyl-CoA is hydrolyzed to give succinate, shown here. As catalyzed by succinyl-CoA synthetase, the energy given off is actually used as a fuel to phosphorylate GDP and thereby make a molecule of GTP. Now this reaction mechanism is really cool. Succinyl-CoA synthetase's active site has a histidine residue, shown here, that's responsible for the full conversion. Now here's the mechanism, which I'd love to analyze on the dog cam if I had one here. Unfortunately, I do not. So what we have to see is a pyrophosphate being brought in. It attacks the carbonyl carbon's electrons. Electrons go up and down and hydrolyze or kick off the thiol group of coenzyme A. At this stage, the histidine comes into the phosphorus center, pushing the electrons up onto the oxygen to release our succinate. The histidine residue of the enzyme is then restored by having ADP presumably come in here and be phosphorylated to ATP. I have to confess that I'm somewhat confused by this, because in the previous slide, which I stole directly from our book, it shows GDP being converted into GTP instead of ADP being converted to ATP. In any event, the transfer of a phosphoryl group, such as this one, from one substrate molecule to another is called substrate-level phosphorylation. In effect, this phosphoryl group is being transferred initially from succinyl-CoA and through on to ATP. In step 6, succinate, which we just formed, is converted over a few steps back into oxaloacetate, which is our starting point for the whole cycle. This reaction is catalyzed by an enzyme called succinate dehydrogenase. The first step, which is an oxidating conversion of succinate to fumarate, you can see it's an oxidation because we remove 2 hydrogens to form a carbon-carbon double bond, is accompanied by the reduction of FAD to FADH2. At this point, fumarate is then hydrated to form this intermediate called malate. Malate then gets oxidized up. You can see that this carbon right here only has 1 bond to oxygen, and it gets oxidized to have 2 bonds to oxygen. In that process, the conversion of malate to oxaloacetate, which is an oxidation reaction on the part of malate, NAD plus gets reduced to NADH. So that brings us back to the beginning. In essence, this is a summary of the entire citric acid cycle, beginning from acetyl-CoA, appending it onto oxaloacetate to form citrate, and then going all the way around each step of the cycle that we've discussed. In net, we see that it produces 3 NADHs, 1 ATP, and 1 FADH2. The net reaction of the citric acid cycle is shown here. So once again, the reaction combines pyruvate, which has 2 carbons, and oxaloacetate, which has 4 carbons, to form citrate, which then goes through the whole cycle to ultimately produce 3 NADHs, 1 ATP, and 1 FADH2. Oxaloacetate is regenerated in the end to catalyze another cycle. Now I know you guys might be confused by my use of the word pyruvate here. I want you to remember that pyruvate is the substrate that is used to make acetyl-CoA, which ultimately feeds into the citric acid cycle and combines with oxaloacetate in the first step. A quote that I love from our book says the following, quote, "Evidence is accumulating that the enzymes of the citric acid cycle are physically associated with one another. This close arrangement of enzymes enhances efficiency because a reaction product can pass directly from one active site to the next through connecting channels, a process called substrate channeling. The word metabolon has been suggested as the name for such multi-enzyme complexes." End quote. I think that the complexity of the biochemical machinery that is involved in the citric acid cycle, as well as all the other biochemical metabolic processes that we learn in this class, is just fascinating and amazing, and I hope that you guys appreciate it as much as I do. We learn also from another quote in our book, "The key catabolic function of the citric acid cycle is the production of high-energy electrons in the form of NADH and FADH2. The electron transport chain oxidizes or burns the NADH and FADH2 formed in the citric acid cycle and ultimately results in the generation of 2.5 ATP per NADH and 1.5 ATP per FADH2. One complete turn through the citric acid cycle generates 3 NADH molecules, 1 FADH2, and 1 ATP for a grand total of 10 molecules of ATP. In contrast, anaerobic glycolysis of 1 glucose molecule generates only 2 ATP molecules and 2 lactate molecules." Now you should remember from our last chapter that pyruvate dehydrogenase complex, or PDC, is what converts pyruvate to acetyl-CoA. It shouldn't surprise you that PDC is a major site of regulation for the citric acid cycle. PDC is inhibited when ATP, acetyl-CoA, and NADH concentrations are high. In other words, when we don't need energy. PDC is promoted when ADP and pyruvate concentrations are high, when we do need energy. PDC isn't the only citric acid cycle related enzyme that's regulated. Isocitrate dehydrogenase is stimulated by ADP when more energy is needed, but inhibited by ATP when energy is not needed. Alpha-ketoglutarate dehydrogenase is another site of regulation. It's inhibited when ATP, succinyl-CoA, and NADH concentrations are high. We'll now finish by reading the last paragraph on page 338 of our text. I wish so badly that I could just show it to you guys on the dot cam. Unfortunately, I'm sitting here in my hotel room recording this, so I'm going to have to just read it to you guys. I think it's a fascinating paragraph. It says, quote, "The use of isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase as control points integrates the citric acid cycle with other pathways and highlights the central role of the citric acid cycle in metabolism. For instance, the inhibition of isocitrate dehydrogenase leads to the buildup of citrate because the interconversion of isocitrate and citrate is readily reversible under intracellular conditions. Citrate can be transported to the cytoplasm where it signals phosphofructokinase to halt glycolysis, and where it can serve as a source of acetyl-CoA for fatty acid synthesis. The alpha-ketoglutarate that accumulates when alpha-ketoglutarate dehydrogenase is inhibited can be used as a precursor for several amino acids and purine bases." The citric acid cycle doesn't just serve to make energy-rich molecules like ATP, NADH, and FADH2. Intermediates in the cycle can also be used in biosyntheses. For example, most of the carbon atoms in the heme groups of hemoglobin and myoglobin come from succinyl-CoA. Also, many of our amino acids are made from alpha-ketoglutarate and oxaloacetate. As we've seen, oxaloacetate is used as a recyclable intermediate in the citric acid cycle. However, oxaloacetate still needs to be replenished from time to time because it sometimes gets siphoned off to make glucose, keeping in mind that the second step of gluconeogenesis, which we talked about back in chapter 17, is the conversion of oxaloacetate to phosphenylpyruvate, as shown here. Looking back at gluconeogenesis, we can see where that occurs, oxaloacetate being converted into phosphenylpyruvate right here. So where do we get more oxaloacetate from once it's used up? Well, you might remember that the first step of gluconeogenesis is the conversion of pyruvate to oxaloacetate. Thus, when we need more oxaloacetate for the citric acid cycle, our bodies can get it by taking glucose through glycolysis to make pyruvate, and then converting that pyruvate into oxaloacetate. So when it's needed, oxaloacetate can be synthesized from pyruvate by using the required steps from gluconeogenesis. Oddly enough, animals do not have the ability to reversibly convert acetyl-CoA back into glucose. Plants and some microorganisms do, however. They accomplish this feat by using a pathway called the glycolysis cycle, which is performed in organelles called glyoxysomes. This is the glycolysis cycle shown in NAD. Now, one thing that I need you guys to understand that can be confusing is this. The purpose of the glycolysis cycle is to convert acetyl-CoA, which plants and other microorganisms have the ability to make or obtain from fats, and in fact so do we, to make a net increase of oxaloacetate. Now you might look at this cycle and say, now wait a minute, don't we have the ability to do that with the citric acid cycle? As it turns out, the answer is no. If you analyze very closely the citric acid cycle with sort of the point of view of an accountant, you will see that for each acetyl-CoA that's taken through the citric acid cycle, there's a net consumption of 2 carboxyl groups, which means that there's not a net production of oxaloacetate. In other words, one acetyl-CoA has gone through to form a catalytic amount of oxaloacetate, which is then regenerated with each cycle, but you don't have a net gain of oxaloacetate. This glyoxylate cycle, which only exists in plants and some microorganisms, bypasses those 2 decarboxylation steps, thereby allowing acetyl-CoA to be converted into a net gain in oxaloacetate. The oxaloacetate can then be siphoned through gluconeogenesis to make glucose. Gluconeogenesis is not shown here, of course. So once again, the purpose of the glyoxylate cycle is to allow plants and some microorganisms to take acetyl-CoA that they obtain from fats instead of from glycolysis and use it to make a net increase of oxaloacetate, which can then be siphoned through gluconeogenesis to make a net increase of glucose. Thus, plants and some microorganisms have the ability to convert acetyl-CoA back into glucose, whereas we humans and other animals cannot do that. Every time we have an acetyl-CoA, it goes through the citric acid cycle. We only make a catalytic amount of oxaloacetate and other byproducts instead of making a net increase in oxaloacetate. I hope that makes sense. I really don't care if you guys know all of the details about this cycle. I just want you to know why it exists and what it does. So that brings us to a conclusion of this, our second and final lecture video for the week. I hope you guys miss me a lot. I miss you. I look forward to seeing you guys when I return from my trip to Texas next week. Until then, enjoy.